angiosperms are much fleshier, flatter, and more leaf-like than most other stamens of living species. Also, the sporogenous tissue is rather massive, similar to that of cycads and cycadioids. Ostrobilea, Himantandra, and Degeneria are unfamiliar to most students. They are plants of South Pacific islands. Figure 23. Two carpels are megasporophylls, therefore, we expect the most relictual living carpels to be rather leaf-like, somewhat resembling megasporophylls of cycads. Several living angiosperms do have carpels resembling young leaves that have failed to open. The two halves of the blade are pressed together, a rather large amount of vascular tissue is present, and a stigma and style are absent. Hairs along the blade margin act like a stigma. If such a carpel produced ovules only at the base and if the upper part elongated, it would begin to resemble a more derived, modern carpel. In the transition from their ancestral group to being angiosperms, fertilization evolved such that the second sperm cell of the pollen tube fuses with the polar nuclei of the megagametophyte, producing the endosperm nucleus. This process of double fertilization is universal in flowering plants. Within the vegetative body, the major transitions were the evolution of vessel elements and sieve tubes. Leaves became broader and developed reticulate venation, and they became more polymorphic, adaptable to a variety of functions, not just photosynthesis. Structural modifications are most easily seen and studied, but probably a more fundamental transition was the acquisition of developmental plasticity, the capacity to survive mutations that alter growth and development. Conifers lack developmental plasticity. For example, most people never have trouble recognizing an unfamiliar plant as being a conifer when they see one because all conifers greatly resemble each other. They are all large or giant trees with one or a few main stems and needle or scale-shaped leaves. They are always green or brown, never brightly colored except for the red seeds of yew and the gray-blue berry-like cones of junipers. This uniformity is even more striking when one considers that gymnosperms dominated Earth for hundreds of millions of years and could have diversified into almost any habitat. On the other hand, angiosperms have tremendous developmental plasticity. They have diversified so greatly that not only hundreds of thousands of species, but also thousands of types, now exist. They range from gigantic to tiny, woody to herbaceous, perennial to biennial to annual, temperate to tropical, desert habitats to mesic to rainforest to aquatic, autotrophic to partially parasitic to fully parasitic, and epiphytic to subterranean to endophytic. Figure 23.3. In many angiosperms, complex compatibility interactions occur between either the pollen and stigma or the pollen tube and stigma style. In some cases, if the stigma and style detect an improper pollen tube, they deposit callose around it and inhibit its growth. The callose block is visible here as a white sheath. Figure 23. For this dwarf daisy is by no means the smallest flowering plant. Many species of mustard, often the first flowers of spring, are also tiny. Umbrophytum subterraneum is a parasite on roots of surrounding plants. The plant consists of an irregular, lumpy tuber and two inflorescences with tiny pink flowers. The root and stem base of the host are visible. Many angiosperms, such as this hydrilla, have adapted to living submerged in water, where they need little sclerenchyma and have no transpiration. All the fruits and vegetables in this market were harvested from angiosperms. Almost all the plants we eat are angiosperms, and, in addition, most of our domesticated animals eat angiosperms such as hay, oats, and corn. The evolutionary changes involved in the conversion of a gymnospermous line into an angiosperm line did not occur instantaneously, nor did they occur in all species. Double fertilization was probably the first transformation because it is universally present. It also occurs in some neophytes, and we are not certain whether it is a shared ancestral feature, indicating that flowering plants and neophytes belong to the same clade or whether it is independent, convergent evolution in two unrelated groups. The ability to produce bisexual flowers must also have been one of the first transformations because all flowering plant clades, including basal angiosperms, have megasporophylls located above microsporophylls on a single axis. Although many angiosperm species produce either staminate or carpellate flowers, we are certain that in every case this has been a loss of parts from a bisexual ancestral flower. Vessel elements possibly evolve next because only one or two basal species lack them. This has been a conundrum for many years. Quite a few plants were considered to be basal angiosperms on the basis of having wood without vessels. It was thought that these were primitively vesselless. Many other features, however, were inconsistent with the idea of these being a natural, early group, and the current hypothesis is that perhaps only Amborella is primitively vesselless and that the others arose after vessels had originated but then these groups lost them. If so, they are secondarily vesselless, and their tracheid-based, gymnosperm-like wood is a derived feature that looks like a primitive one and misled us for years. Sieve tubes may have originated next. Several species still have sieve cells in their phloem. All gymnosperms and seed ferns are or were woody plants and so are most of the basal angiosperms and eudicots. Furthermore, wood in basal angiosperms has many gymnosperm-like features. Therefore, we are confident that ancestral flowering plants were woody perennials. Many extant angiosperms are herbs, as are water lilies and peppers in the basal angiosperms. However, we are certain that this has resulted from the loss of the vascular cambium in these lines, and we are certain this has occurred repeatedly in many clades. The annual habit, the ability to germinate, grow, reproduce, and then die all within a single year, is a uniquely angiosperm feature. No other group of seed plants can do this. Being an annual requires that plants produce flowers shortly after germination, during a period when most gymnosperm plants would still be producing a seedling body with juvenile features. Seedlings and adults often differ strongly in their body characters, and this ability of plants only a few months old to flower may be an important factor in angiosperms' developmental plasticity. Just because a plant has flowered does not mean that it has to die. After seedlings develop the capacity to flower, they could continue to live and grow large, retaining seedling features rather than developing adult ones. Other derived features are the fusion of the carpels into a single structure, fusion of petals into one structure, and floral zygomorphy, that is, flowers that are bilaterally symmetrical, not radially symmetrical. Even more specialized characters such as succulents, parasitism, epiphytism, bulbs, corms, tubers, tendrils, and insect trapping leaves originated only much later, after angiosperms had diversified into many clades. Consequently, each of these innovations is present in only a few families or species. It should not be thought that the flowering plants are now finished, that all their evolution has already happened. Some groups do seem to be changing very little now, but others, especially grasses, composites, bromeliads, and orchids, are still changing and evolving so rapidly that it is difficult to keep up with them. Similarly, we have no reason to expect that the flowering plants will always be the most derived group. Lycophytes dominated Earth for millions of years, only to be displaced by gymnosperma seed plants, which in turn are now being overshadowed by flowering plants. Changing concepts about early angiosperms. Concepts about the nature of early angiosperms have changed as our knowledge of existing and fossil plants has become more complete. In the last century, wind-pollinated trees were grouped together in a subclass Hamamelidae, and were considered the most relictual living flowering plants. This seemed reasonable because these species tend to be large trees with dense wood, their flowers are small and simple, usually without sepals and petals, and many gymnosperms have similar features. After Hamamelid wood anatomy was studied, however, it became obvious that they could not be relictual because their wood contains vessels, fibers, and abundant parenchyma, features not found in gymnosperms. Furthermore, Hamamelid flowers are simple through specialization and reduction. Wind-pollinated flowers do not need to attract pollinators, and thus, they do not need to be large or colorful. they do not need petals or sepals. Recent DNA studies indicate that this syndrome of large, wind-pollinated trees is a derived condition within angiosperms, and furthermore, it has evolved several times in various clades. This is an example of homoplasy, of convergent evolution. And the Hamamelid taxa are now separated from each other and placed among the clades we believe to be their true relatives. Table 23:1 Relictual and derived features in division Magnoliophyta. Approximately 100 years ago, C.E. Bessey developed the hypothesis of the Ronnaline flower, in which a magnolia-type flower was thought to be relictual. Such a flower is generalized, that is, it has all parts, and these are arranged spirally. Also, carpels occur in a superior position, above the other parts. It is easy to postulate the evolution of all of the various existing flower types from a Ronnaline, generalized ancestor. For example, monocot flowers often have 3 stamens and 3 carpels, whereas eudicot flowers usually have 4 or 5 of each. Either condition could arise by a simple reduction of the large number present in a Ronnaline-type flower. Also, the staminate or carpelate imperfect flowers of dioecious species could also arise easily by loss of parts. Figure 23:5 Catkins of pecan. The axis bears many flowers, all with stamens but not carpels. Other flowers on the same plant would be carpelate and give rise to pecan fruits after being pollinated. A flower of magnolia is considered the typical Ronnaline flower. All parts are rather massive and are not specialized for one particular type of pollinator. Very few flowers have so many carpels, almost always fewer than 10. In contrast, seed cones of conifers and cycads have large numbers of megasporophylls. Each carpel secretes a drop of liquid that catches pollen grains, much as conifer megasporangia do. Most botanists had long ago concluded that angiosperms are monophyletic. Features as complex as double fertilization, flowers, and developmental plasticity probably did not evolve more than once. Almost all recent DNA studies also indicate monophily of angiosperms. Currently, many paleobotanists and taxonomists believe that the transition from gymnosperm to angiosperm occurred during the Jurassic and Lower Cretaceous periods of the Mesozoic era. The earliest leaf fossils definitely considered to be those of angiosperms are from the Lower Cretaceous period, approximately 130 million years ago. They represent both dicot and monocot leaves, for example Magnolia phyllum, Phycophyllum, Vitiphyllum, and Plantagenopsis. These fragments are rare in such old rocks, constituting only approximately 2% of all fossils present. In rocks about 50 million years more recent, from the Upper Cretaceous period, many more types of angiosperm leaf fossils are present, outnumbering the leaf fossils of gymnosperms and ferns. These Upper Cretaceous leaf fossils originally were assigned to extant genera such as Populus, Quercus, and Magnolia on the basis of overall shape and size, but careful examinations of fine venation, cuticle, and stomata often show that these fossils are not truly part of modern genera. The original misidentification of these leaves contributed to the erroneous hypothesis that angiosperms must have originated in the Jurassic period, become abundant enough to leave a few fossils in the Lower Cretaceous period, but then suddenly, in the short span of 30 to 50 million years, rapidly diversified into most of the major extant families and even many of the modern genera. After this supposed burst, genera such as Populus, Quercus, and Magnolia would have had to remain relatively unchanged for tens of million years, up to the present. Such a burst of diversification followed by extreme stability did not seem plausible. With the discovery that the Upper Cretaceous and Tertiary leaf fossils only resemble leaves of modern genera but are not identical to them in subtle features, a much more gradual evolution can be assumed to have occurred. The early angiosperms were well established by the Lower and Mid-Cretaceous periods, but probably no modern genus was yet in existence, only precursors that greatly resembled modern genera. These would have evolved gradually into true poplars, oaks, magnolias, and so on. The oldest wood that seems to be from an angiosperm comes from the Aptian epoch. of Japan, unequivocal angiosperm would occurred approximately 120 million years ago. Flowers and fruits appear for the first time in the Lower Cretaceous period, which must mean that their forerunners evolved in the Jurassic period, about the same time that dinosaurs were flourishing. In the last several years, China has explored for fossils much more vigorously, and rich new fossil beds are yielding many new specimens of early angiosperms. Often the fossils consist only of abscised carpels or carpels attached to a receptacle. Unfortunately, sepals, petals, and stamens often abscise even while a flower is still on a plant, and as a result, they are rarely present with the carpels. Perianth parts and stamens of modern plants also tend to be delicate and to decay quickly rather than fossilize. The same may have been true of the early flowers. Pollen that could have come from either relictual dicots or monocots is found in Lower Cretaceous strata. Figure 23:6 A leaf of Magnolia phyllum showing strong similarities to the leaves of living magnolias. However, fine details visible by microscopy show that this is not identical to a true magnolia leaf. Figure 23:7 Part of a flower from the Cretaceous period. Numerous spirally arranged carpels were still attached at the time it died and started to fossilize. The spiral arrangement is similar to that found in flowers of magnolia and water lily. Below the carpels are spirally arranged scars where structures abscised. We do not know if they were sepals, petals, or stamens. Part of a fossil fruit, Peroriomunia puriorensis, approximately 60 million years old. It was a winged fruit similar to those produced today by members of Juglandaceae, the family of walnuts and hickories. Fossil of a hydrangea flower, 44 million years old. This flower is much more modern and derived than that of, having fewer parts. Much attention is now being given to gymnosperms of the Jurassic and Triassic periods, and the focus is centering on cycadophytes and glossopterids. As more is learned about this group of gymnosperms, we realize how much the various groups had begun to develop angiosperm-like features. As you read the following discussion about flowering plants, it will be helpful to study a timeline chart on the evolution of true plants. Classification of flowering plants. Magnoliophyta is such a large group with so many families, genera, and species that it is rare for an individual taxonomist to attempt to study and classify the entire group. In the 1980s and 1990s, the most widely used monograph of the entire division was an integrated system of classification of flowering plants by Dr. Arthur Cronquist of the New York Botanical Garden. Since then, cladistic studies involving DNA, biochemistry, and anatomy often proposed phylogenies that agreed well with those of Dr. Cronquist for certain clades but disagreed for others. The areas where the two systems were inconsistent became the topics of more intense scrutiny of all characters, with subsequent adjustments in our concepts of phylogeny and classification. Currently, two widely used reference books are Plant Systematics, a phylogenetic approach by W. S. Judd et al., and Plant Systematics by Michael Simpson. Their approach is the basis for the treatment used here. Also of special importance is the Angiosperm phylogeny group, which is a semi-informal collaboration of many systematists. Their classifications can be found on the website for the Missouri Botanical Garden. Figure 23:8 A reconstruction of a Glossopteris, a possible relative to the ancestors of flowering plants. The leaves had a prominent midrib and reticulate venation. The leaves were deciduous, and the wood had annual rings, indicating that it lived in a temperate climate. Figure 23:9 recently proposed cladogram of angiosperms. Basal angiosperms are Ambarella, nymphiales, Austrobaleoles, and magnoliids. The origin of Chloranthaceae, Ceratophyllum, monocots, and eudicots is an unresolved polycotomy. Soon after their origin, flowering plants began to follow two distinct lines of evolution, and currently, almost all angiosperms are classified as monocots or eudicots. No single character always distinguishes a monocot from a eudicot, and some species would fool most botanists. In general, monocots, as their name implies, have only one cotyledon on each embryo, and other typical characters are the following. Their leaves usually have parallel veins because the leaves are elongate and strap-shaped. Vascular bundles are distributed throughout the stem, not restricted in one ring. And monocots never have ordinary secondary growth and wood. Flowers of monocots have their parts arranged in groups or multiples of 3 sepals, 3 petals, 3 stamens, and 3 carpels. Eudicots are much more diverse and include a greater number of families, genera, and species. Eudicots have 2 cotyledons and reticulate venation in the leaves. Vascular bundles occur in only one ring in the stem. They can be woody, herbaceous, or succulent or have any of many highly modified forms. Flower parts occur in sets of 5 most often and sometimes in sets of 4, but they only rarely occur in sets of 3. A surprising result of cladistic studies is that the monocot-eudicot divergence did not occur right away, but instead, the early angiosperms diverged into several clades now called the basal angiosperms. These are not newly discovered species, it is just that we had classified them as monocots or dicots before, but many different DNA sequences indicate they had become reproductively separate from the other angiosperms very early. Of the basal angiosperms, Amborella and Austrobaleoles greatly resemble eudicots. However, they cannot be classified with the eudicot clade because the common ancestor that includes both them and the eudicots also includes monocots, and we cannot construct a group that includes some descendants of a common ancestor but excludes others. Similarly, nymphaeals have many features of monocots, but classifying them as monocots would create a polyphyletic group. Figure 2310 lilies and their relatives are excellent for demonstrating the typical monocot flower. They have 3 of each type of appendage and all are large enough to be seen easily. The 3 carpels are fused together, but a transverse section would show 3 chambers. 1 petal and 2 stamens were removed to reveal the ovary. Plants and people. Box 23-1 Maintaining genetic diversity. The flowering plants are particularly adept at evolving rapidly. In the short time they have existed, they have diversified greatly. The result is that hundreds of thousands of species exist, each differing from the others in features such as flowers, fruits, vegetative structure, and metabolism. Within this panoply of diverse plants, we and our ancestors have discovered a wealth of foods, medicines, and soul-soothing flowers. Now, at the beginning of the 21st century, we have become aware of the horrific ecological destruction occurring on Earth, with vast areas of vegetation being cut or burned or flooded with artificial lakes behind hydroelectric dams. We have also begun to realize that we are causing the extinction of hundreds of species every year and, in the process, their genetic information is being lost. On a philosophical basis, we can debate whether one species, Homo sapiens, has the right to destroy so many other species, but on a more pragmatic basis, we should consider whether we are unwittingly destroying potential sources of new foods and medicines. Consider just 2 examples. A relatively unknown species, quinoa, can be grown in relatively dry regions with poor soil, yet it produces an abundance of nutritious, protein-rich seeds. And Pantodiplandra brasiliana produces a small protein that is 2,000 times sweeter than sugar. This could possibly be used as a completely natural, safe, nutritious, low-calorie sweetener, decreasing the amount of sugar or artificial sweeteners that we consume. How can botanists preserve the genetic resources of the plants, animals, and microbes that coexist with us? Several types of efforts are needed, and 2 particularly important ones are habitat preservation and germplasm banks. Germplasm banks were started several years ago as rather simple seed storage facilities, where bags of many types of seeds were maintained under cool, dry conditions. After several years, while most seeds were still healthy, they would be germinated, grown into mature plants, and then their fresh, healthy seeds would be collected and stored. After several years, the process would be repeated. This is still done for many species, but now the seeds can be kept for extremely long periods, held at very low temperatures or even in liquid nitrogen. Furthermore, it is now possible to maintain some seeds as tissue or cell cultures, either grown slowly in culture tubes or kept as frozen cells. In the foreseeable future, it may be that some plants, or at least some of their genes, will be kept simply as DNA molecules in solution or in cloning vectors. Currently, there are over 100 germplasm banks throughout the world, preserving at least 3 million types of plant material. Examples are the Svalbard Global Seed Vault, built in an abandoned coal mine in the permafrost of northern Norway, and the National Seed Storage Laboratory located at Colorado State University and run by the U.S. Department of Agriculture. The NSSL was established in 1958 and has more than 232,000 seed types. At present, its size is being quadrupled, and its capacity will be more than 1 million seed samples. The Millennium Seed Bank project at Kew Gardens in England reached its goal of safeguarding 25% of all the world's plant species in October 2020. The purpose of germplasm banks is not merely to store and maintain plant genetic diversity but also to make it available. Botanists can request samples and use them for research and plant breeding programs. If some are found to have useful traits, they can be grown as crops. Of course, it is also hoped that in many cases we can use these plants to help restore or rebuild ecosystems that we have already damaged. Basal angiosperms. The basal angiosperms contain the living descendants of several groups that originated while angiosperms were still a young clade. Their ancestors became reproductively isolated from the other early flowering plants before distinctive angiosperm traits had originated. It would be easy to assume that the descendants of the three earliest diverging groups would all display a large number of primitive traits and few derived ones. They should look primitive, and they should look like taxa that had just barely become distinct from some gymnosperm sister group. All organisms evolve, however, and so did the descendants of the basal angiosperms. The three groups of extant descendants of these clades, Amberlaceae, Nymphaeaceae, and Austrobaleales, have not remained static evolutionarily and have not preserved all ancestral features intact. One group should be familiar to you. Nymphaeaceae are the water lilies. They are small, soft-bodied herbs with vascular bundles scattered like those in monocots, and they completely lack any wood. In most, their stems must be submerged underwater, and exposure to ordinary conditions on land would kill them. Their large colorful flowers are pollinated by animals. They have almost nothing in common with either gymnosperms or seed ferns. Amberella trichopoda is very different, being small trees in forests of New Caledonia. Their wood contains tracheids but no vessels and little parenchyma. This species is dioecious, some plants having staminate flowers, others having carpellate flowers. Both flowers are small with 5 to 11 tepals. Staminate flowers have numerous stamens, and carpellate flowers have 5 or 6 carpels whose edges do not seal tightly but instead have interlocking, secretory hairs that act as a stigma. Austrobaleales contains woody trees with bisexual flowers, with stamens and carpels being similar to those of Amberella. Figure 23:11 flowers of water lilies have numerous sepals, stamens, and so on, with all parts arranged in spirals like the leaves of a rosette plant. This is believed to be a relictual feature similar to a gymnosperm cone. Monocot flowers would be derived from an ancestral flower like this by mutations that result in only 3 or 6 of each type of floral appendage. Amberella. Because water lilies, Amberella, and Austrobaleales differ in so many features, they cannot all resemble the earliest angiosperms. Instead, one or the other, or all three, have undergone considerable evolutionary change since their clades originated. Water lilies have little in common with either gymnosperms or seed ferns, almost certainly many of their features have changed considerably. Amberella and Austrobaleales appear to have changed little, but it is important to realize that they have not remained static. We cannot assume that every feature of these woody trees is like that of early angiosperms. Instead, we assume that features present in them and gymnosperms are relictual features, plesiomorphes. Ancestors of the magnoliid clade diverged from early angiosperms a bit later than Ambarella and Austrobaleales, but this conclusion is based mostly on DNA evidence. Morphologically, the magnoliids do not differ greatly from these two earlier clades. For example, family Magnoliaceae contains trees with wood similar to that of gymnosperms in that it lacks vessels, fibers, and axial parenchyma. Magnolia flowers have numerous stamens and carpels arranged in spirals. Furthermore, carpels are not fused together into a pistil as occurs in almost all monocots and eudicots. Another important feature is that their pollen grains have only a single germination pore. They are unioperturate, as are all other basal angiosperms and monocots. Eudicots have 3 germination pores. Other magnoliids are laurels and avocado and peppers and peperomias. Table 23. 2 representative families of the magnoliid clade. Figure 23:12. All early fossil pollen has only one aperture, but almost all living eudicots with many derived features have 3 apertures. Therefore, unioperturate pollen in living angiosperms, such as this Siphonoglossa, is considered a relictual feature. Pollen of Lophospermum has 3 germination pores. The pores are located in grooves, something that does not occur in any early fossil pollen. Monocots. Currently, monocots are widely believed to have arisen from early angiosperms. Approximately 80 to 100 million, perhaps even 120 million years ago. Because all monocots lack ordinary secondary growth and wood, their ancestors were probably herbs with either no vascular cambium or little cambial activity. The gynoecia of many monocots are composed of several carpels that are either free of each other or at most only slightly fused together. The perianth usually consists of 3 outer and 3 inner members. They often look so similar that rather than using the term sepals and petals, the perianth members are just called tepals. Monocot tepals rarely fuse to each other to form a tube. Instead, they remain free. These features must have been present in the group from which the monocots evolved. Water lilies have similar features. An early set of angiosperms may have resembled nymphaeals and may have given rise to both the water lilies and the monocots. One theory about the parallel venation of monocot leaves postulates that the ancestors had broad leaves and lived as aquatic plants. The leaves evolved to a more reduced, simple type without a blade, a form more adaptive for a submerged leaf. Some of these plants moved into drier habitats where their leaves were not submerged and a broad or long lamina would be advantageous. Mutations that resulted in a basal meristem were selected, resulting in strap-shaped leaves. In the evolutionary lines of broadleaf monocots such as palms, philodendrons, and dieffenbachias, a marginal type of meristem evolved. It was located at the end of the residual leaf, resulting in the formation of what appears to be a petiole and lamina. Early monocots diverged into a series of clades whose extant members are classified into approximately 10 orders. Alismatales. Alismatales contains many aquatic herbs such as Sagittaria and many aquarium plants such as Hydrocharis, Najas, and Hydrilla. These plants are most often found in swamps and marshes, partly or entirely submerged. Although many species retain a large number of plesiomorphic features, others have become highly modified in response to the unusual selection pressures associated with an aquatic habitat. Plants of Hydrocharotaceae, Najadaceae, Posidoniaceae, Rupiaceae, and Zosteraceae grow completely submerged in seawater and are known as sea grasses. They have no transpiration, and thus, mutations that result in the loss of stomata are not selectively disadvantageous. Air chambers make the plants buoyant, therefore, mutations that prevent the formation of lignified fibers are selectively advantageous. The plants do not waste carbon and energy by producing fibers that are not needed for support. Such plants tend to be thin and delicate, having very little sclerenchyma and almost no xylem. Tissues have been lost or simplified, the ancestors of elismatales were more complex and massive and had more sclerenchyma. Members of Elismataceae have a part of their bodies emergent above the water's surface, and some are entirely emergent whenever their marsh habitat dries temporarily. These plants are less highly modified, often having large leaves, considerable amounts of fiber, and a thick cuticle on the leaf epidermis. It is necessary for a species to be adapted to the most adverse conditions that it frequently encounters during one plant's lifetime. Figure 2313 monocots are believed to have originated as semi-aquatic plants that inhabited swamps and marshes. The leaf may have consisted only of a leaf base and petiole, the entire blade having been lost evolutionarily. A pseudolamina evolving from a petiole. A broadleaf evolving from a petiole. Figure 2314. Some monocots, such as this banana, have broad leaves, but their lamina is not homologous to that of a dicot leaf. In this monocot broad leaf, all vascular bundles run parallel to each other from the midrib to the margin, whereas in a dicot lamina, they diverge and branch into a reticulate pattern. Figure 2315. One recently proposed cladogram of monocots. There are many unresolved polycotomies. Figure 2316. Sagittaria, a member of elismatales, is dioecious. Plants have either staminate or carpellate flowers. Table 23.3. Representative families of elismatales. Figure 2317. This anthurium is an aroid, one of the monocots that have a eudicot type of broad leaf. Compare its reticulate venation with the parallel venation of banana. Many members of this family are adapted to conditions of low light and high humidity. Most are members of tropical or subtropical habitats. Araceae have a characteristic inflorescence with a sheath-like bract surrounding a thick column. Tiny staminate flowers are embedded in the top of the spadix, carpellate ones in the base. Peltandra virginica. In the most relictual members of elismatales, flowers are large and showy, with 3 sepals and 3 petals, but in others, they are highly modified, especially in those species in which the flowers do not emerge from the water and pollination occurs below the surface. Such flowers often completely lack a perianth. Family Araceae contains numerous familiar houseplants, philodendron, anthurium, and ericema. Dieffenbachia and 1,800 other species also belong here. This family is characterized by the evolution of a distinctive inflorescence, tiny flowers, either unisexual or perfect, embedded in a thick stalk, the spadix. Staminate flowers are located near the top of the spadix, carpellate flowers near the base. The spadix is surrounded by a large bract, the spatha. Lilialesh. In the past, Lilialesh was defined broadly as a large group with many highly derived families. It was basically defined as the petaloid monocots, those with large, colorful flowers. Recent DNA evidence indicates that Lilialesh is a smaller clade with approximately 11 families and 1,300 species. Many of its previous families are now grouped in asparagales. Important features in Lilialesh are the presence of spots or lines on the petals and of rather ordinary nectaries formed at the bases of tepals or stamens. Flowers in asparagales typically have septal nectaries. One of the largest families, Liliaceae, contains so many ornamental plants, mostly bulbs, that most people are familiar with them and think of them as the typical monocots. Liliaceae contains lilium, tulipa, calochortus, and fritillaria. Colchicaceae contains gloriosa and colchicum, the source of the alkaloid colchicine that prevents microtubule polymerization. Perhaps the most unusual member of Lilialesh is Smilacaceae, with the single genus Smilax. Despite being closely related to lilies, these are tough, fibrous vines with leaves that have a petiole and broad lamina with reticulate venation, not lily-like at all. At first glance, it is easy to assume they are dicots. Asparagales. This is a large clade with many families, species, and types of biology. By examining this clade, we get a sense of evolution as diversification. There are several morphologic and DNA synapomorphies that unite this group. Most form nectaries in an unusual way. Their carpels fuse side by side starting at their bases, the fused sides being called septa. They do not fuse all the way up to the style, however, and the open areas secrete nectar. These are septal nectaries. Beyond this, the group is extremely diverse in morphology, ranging from small, delicate bulbs like hyacinth, chives, and onion to vining epiphytes such as many orchids. Bulbs, rhizomes, and corms are common. Agaves and yuccas have giant, fibrous perennial leaves. Iris has flattened sword-shaped leaves, and many members such as narcissus have delicate leaves that live only during spring and summer. Several members of Agavaceae and Ruscaceae have anomalous cambia and grow to be large, highly branched trees. Asparagales has diversified biochemically as well, with different groups having distinctive chemical compounds, such as the sulfur compounds in onion and garlic or particular alkaloids in Amaryllidaceae. Flowers are large, colorful, and showy for the most part, and in Orchidaceae, numerous types of insect pollination have evolved, many accompanied by distinctive aromas. Table 23. 4 representative families of asparagales. The Orchidaceae is the largest and most diverse family of asparagales. The most familiar, ornamental ones are epiphytic, but many are terrestrial and one is a subterranean parasite. Orchid flowers are highly modified from the ancestral conditions of angiosperms, being zygomorphic with complex shapes, colors, and fragrances that attract specific pollinators. Orchids are unusual in producing hundreds or thousands of tiny seeds in each fruit. The seeds are dust-like and so undeveloped at germination that they must form a symbiosis with certain fungi in order to survive long enough to form roots and leaves. Figure 23. 18 members of asparagales. Iris flowers appear to have many unfamiliar parts, but the uppermost petaloid parts are actually stamens, the long dark regions are sporogenous tissues, the anthers. In many angiosperms, structures that appear to be petals are really sterile stamens. Like bamboos and palms, yuccas and agaves prove that herbs do not have to be soft and small. The leaves are long and narrowly triangular with parallel venation, and they may be thick at the base. They are arranged as a rosette around a short or long stem. Most have a needle-sharp point and razor-like hooked spines along the edges of the leaf. Dioscoriales. This small order has only one family, Dioscoriaceae, and is mentioned because it has a familiar, important food crop and unusual morphology. Yams are starchy tubers produced by several species of Dioscorea and are a major source of carbohydrates for many people of tropical areas. Also, Dioscoreas have petiolate, broad leaves with reticulate venation and are easily mistaken for dicots. Commelinoid monocots. The following four orders of monocots are known as the commelinoid monocots because they differ from the others in several unusual synapomorphies. They have unique types of epicuticular wax. Their walls have unusual types of hemicelluloses and ultraviolet fluorescent compounds. Their pollen contains starch, as does their endosperm. Multiple studies of RBCL, ATPB, and other DNA regions support this as a distinct clade. Arachales. This order contains familiar plants, the palms, in family Ericaceae. There are about 3,500 species, all of which are easily recognizable by their solitary trunk, which varies from 1.0 m wide in some and only 1 cm in others. All have scattered vascular bundles, which is typical of monocots. Leaves of palms always occur only near the shoot apex, never distributed along the length of the stem. In a few species, trunks are prostrate. Or vines. All species have simple leaves that, after fully expanded, are torn by wind into either a pinnate pattern or palmate one. Coconuts and dates are two types of palm fruit, but many others exist. Palm flowers are seldom seen because they are usually tiny, approximately 5 mm across, and are formed only high up in the tree. Figure 23:19 Palm flowers usually occur in large inflorescences at the top of a plant. Although palm flowers are small, they have typical monocot organization with their parts in sets of 3. Coriacea. Poles. This order contains the grass family Poaceae as well as several other familiar groups such as cattails, bromeliads, and sedges. Poaceae contains about 8,000 species and are much more than just the plants in the lawn. They also include most foods, such as wheat, barley, oats, rye, corn, rice, and sugar cane, as well as a major building material of the tropics, bamboo. Humans began farming grass crops as early as 10,000 years ago, and approximately 50% of all calories consumed by people come from grass seeds. Also, our main sources of meat, cattle, pigs, and sheep, are raised on grassland and fed corn. Grasses are abundant in flat, open, dry regions in the central areas of all continents, such as the rangelands of the United States and Africa, the steppes of Russia and Ukraine, and the pampas of Argentina. This accounts for 1/4 of all vegetated land on Earth. Some grasses grow as tight clumps, and others spread by rhizomes or runners. Either way, their shoot-apical meristems tend to be located near the ground, protected from fire and grazing animals. As a result, they survive both better than most other types of plants. Widespread grasslands are a new ecologic development, however, arising less than 25 million years ago. During the Miocene epoch, extensive forests disappeared and were replaced by grasslands, making possible an increase in grazing animals and perhaps even in ancestral hominids switching from a life in trees to one on the ground in African rangelands. Table 23:5 Representative families of poles. All grasses are wind-pollinated, so sepals and petals are of little importance and are reduced to bristle-like structures. The 3 carpels are fused together, but in most grasses, the 3 stigmas remain separate. An ear of corn is an inflorescence, and each silk is a compound style and stigma. Within the fused ovary of grasses there is just one ovule. Once fertilized, it matures into a seed whose seed coat fuses firmly to the developing fruit wall. The objects we usually call seeds of corn, wheat, and oats are actually single-seeded dry fruits known as caryopses. Nearly the entire bulk of the seed is endosperm. The embryo is small but well developed. Figure 23:20 Grass flowers are very reduced, simplified, and wind-pollinated. The usual 2 small, dry scales are thought to be remnants of an ancestral bract and 2 fused tepals. The third tepal does not form. 3 anthers with long filaments and 3 carpels fused together with long feathery styles and stigmas are usually present. Grass flowers occur grouped together in complex, compact inflorescences, and inflorescence characters are important for identifying grasses. Closely related to grasses are sedges and rushes. All 3 families contain plants of wet, marshy areas, with tiny, reduced flowers and small dry fruits. Sedges and rushes are often confused with grasses, but sedges have edges, rushes are round, and grasses are hollow from top to ground. Sedge stems are triangular in cross-section. Those of rushes are round and solid, and those of grasses are round but hollow. This simple rhyme is mostly true, but there are exceptions. Other members of poles are cattails and bromeliaceae. Cattails grow in ponds and marshy areas. Each plant spreads by thick, horizontal subterranean rhizomes, with some axillary buds growing out as more rhizomes, and other axillary buds growing upward and producing the familiar green leaves. What appears to be a cattail plant is really just a single branch. Their characteristic reproductive structures consist of a tip with many staminate flowers and thousands of carplet flowers, although by the time we see them, the staminate flowers have usually withered and the carplet flowers have matured into tiny fruits. Bromeliaceae contain some of the most beautiful tropical epiphytes, their large, brightly colored inflorescences being easily visible even in thick jungle vegetation. Epiphytic species extend as far northward as the subtropics, Spanish moss and ball moss occurring from Florida to Texas. Other species are terrestrial and usually xerophytic, such as puya of Chile and Peru and pineapples. These xerophytic terrestrial forms are believed to be relictual, and their capacity to withstand drought may have made it easier for bromeliads to adapt to life as epiphytes. Because bromeliads occur in only the Americas, it is assumed that they evolved after South America separated from Africa approximately 80 million years ago. Had they evolved earlier, numerous species should occur in the African rainforests and coastal deserts. Zingiberals. This order contains some of the most familiar of all houseplants, Maranta, Calathea, canna lilies, and gingers, as well as some that are best known in the warmer southern states, banana and bird of paradise. Members of zingiberals differ from most other monocots in that they tend to have large, showy flowers pollinated by insects, birds, or bats. Furthermore, many of their flowers have derived features. Adjacent sepals are often fused to each other, forming a tube, and the same is often true of the petals. Many flowers are bilaterally symmetrical. The gynoecium typically consists of 3 carpels that have fused almost completely. Very often the gynoecium is inferior, located below the sepals and petals, a condition that is always considered quite derived, the result of considerable evolutionary modification. Figure 23-21. Most sedges grow in wet, marshy areas. Cattail, Typha, has hundreds of staminate flowers above and thousands of carpelet flowers below. As a cattail breaks apart, the tiny one-seeded fruits blow away. Seedlings are minuscule, like short threads. The largest family, Zingiberaceae, has approximately 1,000 species. Almost all are tropical, and most are soft, non-leathery herbs. The leaves are broad and have a petiole. During the evolution of the zingiberals, mutations that resulted in the petiole leaf becoming flattened and broadened were selectively advantageous. The plants grow in the heavy shade of jungle understory where light interception is important. Figure 23-22. Many epiphytic bromeliads catch rainwater with a tank made by their overlapping leaves. They absorb water through their leaf bases rather than through their roots. Their flowers develop underwater. Spanish moss, Tillandsia usnioides, is a bromeliad, not a moss. It grows attached to tree trunks as an epiphyte, not a parasite. This species grows in the southeastern United States, where humidity is high and rainfall is frequent. Many bromeliads have brilliantly colored flowers and bracts, which may make the flowers more visible in the dim light of rainforests. Table 23-6. Representative families of zingiberals. Eudicots. The eudicots constitute a much larger group than the monocots and are divided into numerous clades, some of which have diversified into many families, others having fewer members. Virtually every type of organ, tissue, and metabolism has several or many variations, so they are more difficult to characterize than are the monocots. One especially distinctive feature is that their pollen grains have either 3 germination pores or have some condition derived from the triculpate mechanism. Furthermore, their flower parts are arranged in whorls, not spirals, and their stamens usually have a well-defined filament and anther. They are not flat or leaf-like at all. These characters are greatly modified from those of sporophylls found in other vascular plants. Current cladograms of eudicots have a large number of orders, too many to be covered here. Those that have special importance and that contain plants that you might know are presented below. Figure 23-23. One recently proposed cladogram of eudicots. There are several unresolved polycotomies. Basal eudicots. In older classifications in which all flowering plants were assigned to either monocots or dicots, many families did not seem to fit well in either group. Several, such as water lilies, are now assigned to the basal angiosperms. Ranunculales and several others are believed to be clades that diverged at early stages in eudicot evolution. Ranunculaceae is interesting because its flowers have so little fusion of parts. Each flower usually has many stamens and carpels, all of which remain separate of the others. Stamens may remain free or may fuse together into a tube or even fuse to the carpels. Familiar members of Ranunculaceae that have numerous, free parts are buttercups, windflower, and clematis. The poppy family, Papaveraceae, is well known for its numerous ornamental species such as argemone, Eschscholzia, and papaver. Papaver somniferum is opium poppy, the source of a milky latex harvested for opium, morphine, a strong analgesic that depresses the cerebral cortex, is extracted from opium, as is codeine. Morphologically, poppies are unusual in being either herbs or shrubs with a very soft wood. Several hamamelid families are now believed to be basal eudicots rather than basal angiosperms. These are large trees with reduced wind-pollinated flowers, usually in dangling inflorescences containing many staminate flowers and just a few carpellate flowers. A familiar example is Platanaceae, sycamores. These large trees have small, inconspicuous clusters of flowers in the spring that mature into spherical clusters of hundreds of tiny dry fruits. Caryophyllales. DNA-based studies have combined a large number of families into a group called Caryophyllales. Most of these families had long been known to be closely related and were also known as Caryophyllales. In new treatments, the old group is referred to as core Caryophyllales. Examples of the core group are cacti, isoplant, portulaca, bougainvillea and phoroclox. Spinach, beets, and Russian thistle, and carnations and chickweed. The core Caryophyllales share many derived characters, but one is especially important. Whereas other flowering plants have anthocyanin pigments in their flowers, almost all Caryophyllales instead produce a group of water-soluble pigments called betalains. The ancestral group may have lacked petals in their flowers, a condition common in many Caryophyllales. Although many appear to have petals, we believe that these are modified sepals in some families and modified stamens in others. Figure 23-24 carpels in ranunculales remain free of each other at maturity, they do not fuse during development. Consequently, each flower produces many separate fruits. Clematis. Poppies have numerous free stamens but fused carpels. They have no styles, and thus, the stigmatic surface is the top of the ovary. Another unifying character of Caryophyllales is that endosperm develops only a little and then fails to continue growing. Instead, nucellus cells proliferate and form a nutritive tissue called perisperm, which surrounds the developing embryo. Perisperm is usually absorbed almost completely by the time the seed is mature. A third feature that unites this order and distinguishes it from all other taxa is the nature of sieve tube plastids. In Caryophyllales, phloem plastids contain deposits of fibrous protein located as a ring just interior to the plastid membrane. In other angiosperms, plastids contain either starch or crystalline protein with a different structure. Table 23-7 Representative families of Caryophyllales. Figure 23-25 Caryophyllales, Echinocereus coccineus and Carpobrotus. The brilliant colors in the petals of both are due to betalan pigments. Figure 23-26 Core Caryophyllales are characterized by the presence of betalan pigments in flowers and fruits. There are two basic types, beta-cyanins and beta-xanthins. The Caryophyllales are postulated to have arisen from ancestors similar to Ranunculaceae. Most members of Caryophyllales are herbaceous, with either no wood, very little wood, or unusual, anomalous wood, so the ancestral group is suspected to have been herbaceous or shrubby, having lost the capacity for extended, massive secondary growth. The time of origin of Caryophyllales is approximately 70 to 80 million years ago, at an important time of continental drift. At that time, all land in the southern hemisphere was joined together in a giant continent now called Gondwana. Caryophyllales originated in southern Gondwana, and its earliest families spread across that continent. Then continental drift caused Gondwana to break up, first with South America moving westward and later with Australia, India, and Antarctica moving eastward. Families that had already come into existence were carried along with the moving land masses and are now present on most or all the southern continents. Families that originated after the breakup of Gondwana arose in individual continents and are now mostly confined to those. For example, cacti arose in South America and later spread to Central and North America, but its seeds could not cross either the Atlantic or Pacific Oceans, thus, cacti do not occur naturally on any other Gondwanan continent. Similarly, Didieriaceae arose in East Africa and Madagascar and occur nowhere else. Figure 23-27 in core caryophyllales, endosperm develops for only a short while or not at all and is not sufficient to nourish the embryo. Instead, the nucellus cells proliferate and act like endosperm, forming a tissue called perisperm. Figure 23-28 plastids in sieve tube members are simple, but they may accumulate particles of starch or protein or both. The nature of the accumulated material is highly specific with regard to the family, and phloem plastid analysis is important in studying the evolution of flowering plants. Sieve tube plastids of Monococcus lack a central cubic protein crystal, but those of MacArthurria have one. Both are members of caryophyllales, and as a result, both have a peripheral ring of protein filaments. Several of the other families that are now placed in the expanded caryophyllales are the buckwheats, pokeweeds, and two groups of carnivorous plants, Droseraceae and Nepenthaceae. Santallales. This is a small order of highly modified plants, most of which are parasitic. The sandalwood family contains the large tree santalum from which sandalwood incense is obtained. It appears to be an ordinary tree, but its roots make fine, almost imperceptible connections to roots of surrounding plants and parasitizes them. Also in this family are common mistletoes, viscum and phoradendron, which are used as decorations at Christmas. These mistletoes have chlorophyll and are photosynthetic, therefore, they are just hemiparasitic, but some members are holoparasitic, having no chlorophyll at all. Viscum minimum lives entirely within its host plant, Euphorbia. Loranthaceae is a large family with about 900 species, all of which are parasitic, but none of which occurs in temperate North America. Figure 23-29 mistletoe is a hemiparasite, it carries out some photosynthesis. Its hostorial root system penetrates the host tree branch at the top of the photograph. Rosid clade. The remaining eudicots are members of two very large, very diverse clades, the rosids and asterids. The rosid clade consists of many families that, taken as a whole, are so diverse with respect to vegetative body, flowers, chemistry, and ecology that it is difficult to see they are all related. However, some share enough characters with others to indicate a relationship. And those of the second group share different features with a third group and so on until a larger picture of phylogenetic relationships can be distinguished. DNA analysis has added a great deal of information to identifying patterns of relationships. The rosids consist of several small orders and two large groups. One of the small orders has few members but great economic significance. Vitales contains Vitaceae, the grape family. Vitis is the genus of grapes that give us juice, raisins, and table grapes. Vitis vinifera is the wine grape, and despite the enormous variety of wines, virtually all are made from varieties of this one species. The other small order, Geraniales, contains the geranium family, Geraniaceae. The two large clades of rosids are the fabids and the malvids. These two clades together contain more than 100 families, and it is difficult to give any universal characters. As a group, rosids are more derived than basal tricolpates from which they arose and have derived characters that differ from those of caryophyllales. Rosids are especially interesting in that none of them has any of the highly relictual features found in many basal angiosperms. Table 23 8 representative families of the rosid clade. An important character in rosids is the presence of pinnately compound leaves. This is believed to have been the ancestral condition. Although some living species have simple leaves, these apparently have arisen from compound leaves, perhaps by suppression of all leaflets except one. Whereas simple leaves are an early, ancestral condition for the angiosperms as a whole, they are a later derived condition for rosids. Although this clade is named for the order Rosales, roses should not be considered typical, they are just one group of many. Because the rosids consist of 14 large orders with over 50,000 species, only a few can be mentioned here. 5 of the orders contain almost 75% of the species: Fabalesh, 19,000 species, Myrtales 9,000, Malpighiales, 1,600, Rosales, 6,300, and Sapindales, maples, horse chestnuts, creosote bush, and the species whose resins are valued as frankincense, boswellia, and myrrh, comifera, 5,800. Members of this subclass include roses, of course, and legumes, fuchsia, evening primrose. Dogwood, the spurges that look like cacti and often have an extremely poisonous milky latex, maples, and dill, celery, carrot, parsley, and hemlock. The rose family is important not only in an evolutionary sense but also economically. Rosaceae contains numerous ornamental genera, including Rosa, Crategus, Spirea, Cotoneaster, Pyracantha, Photinia, Potentilla, Cynamales, and Sorbus. The family also provides most of the fruits that can be grown in temperate climates: Malus, Pyrus, Areobatria, Fragaria, and Rubus. Fabaceae is also a large family having bodies that vary from herbaceous annuals to shrubs, vines, and long-lived trees. It is important economically as a source of many foods, drugs, dyes, and woods, and many of its species are the dominant plants in arid areas and deserts. Many species have root nodules with symbiotic associations with nitrogen-fixing bacteria, and thus, they can grow in poor soils yet still produce protein-rich seeds. Legumes are a critically important source of proteins for people in arid areas. Asterid clade. The most derived large clade of eudicots is the asterid clade, which contains plants such as sunflower, periwinkle, petunia, and morning glory. Asterids, being a sister clade of rosids, originated perhaps as recently as 60 million years ago, and even its most basal members were much more highly derived than plants in the basal angiosperms. The majority of asterids can be easily distinguished from other angiosperms on the basis of 3 features: they have sympetallous flowers, they always have just a few stamens, not more than the number of petal lobes, and stamens alternate with petals. Asterids exploit very specialized pollinators that recognize complex floral patterns, and such plants could not evolve before derived, sophisticated insects appeared. Many chemical differences exist between this group and all others. It lacks many specialized chemicals found in other clades. It has no betalanes, benzylisoquinoline alkaloids, ellagic acid, or proanthocyanins. Instead, many asterids have iridoid compounds, which occur only rarely outside this group. Certain families produce numerous, very potent chemicals that deter animals or kill them outright. Apiaceae have secretory canals containing oils and resins, triterpenoid saponins, coumarins, falcarinone polyacetylines, monoterpenes, and sesquiterpenes. The poison Socrates was given to kill himself came from Conicum in the Apiaceae. It is not important for you to learn this list or understand what these chemicals are, but rather to see that in addition to new morphological, anatomical, and reproductive characters originating evolutionarily, new defensive chemicals also arise. Currently, asterids have the greatest number of species, but they are grouped into 2 small orders and 2 groups of orders, lamiids and campanulids. One family contains fully 1/3 of all the species and is the largest family of eudicots. Examples of asterids are milkweeds, potato, tomato, red peppers, eggplant, tobacco, deadly nightshade, and petunia, morning glory, thyme, mints, and lavender. And Asteraceae with sunflowers, dandelions, lettuce, chrysanthemum, ragweed, and thistle. Many asterids are extremely important medicinally. Apocynaceae contains periwinkle, vinca, from which are extracted vinblastine and vincristine, two of our most potent anti-cancer drugs. Rubiaceae contains, in addition to coffee, cinchona, from which we derive the anti-malaria drug quinine. Another rubiaceous genus, Cephalus, provides us with ipecac, an emetic used to induce vomiting in cases of oral poisoning. Heart disease is treated with cardiac glycosides extracted from digitalis. These compounds make heart muscle beat more slowly and strongly, increasing output of blood from the heart and improving circulation. The order Lamiales is quite derived in its floral characters. The flowers tend to be bilaterally symmetrical and of sizes and shapes that permit only certain insects to enter and effect pollination. The floral bilateral symmetry further forces the insect to enter in one particular orientation. Pollinators capable of doing this tend to be sophisticated and able to recognize different flowers easily. They learn which flowers provide the greatest rewards and then search for other flowers of identical shape, color pattern, and fragrance, thus providing efficient pollination for the plant species. The giant family Asteraceae contains 1,100 genera and 20,000 species distributed worldwide in almost all habitats except dense, dark forests. They range from important food plants to ornamentals to weeds. The characteristic daisy sunflower type of inflorescence makes them instantly recognizable. Members of Asteraceae have a wide range of unique chemical defenses against herbivores, sesquiterpene lactones, monoterpenes, terpenoids, and latex canals that contain polyacetylene resins. The presence of these chemicals makes composites extremely resistant to animals that eat plants or lay eggs in them. It also causes them to be irritating to human skin, resulting in numerous cases of contact dermatitis. The Asteraceae is a young family, perhaps no more than 36 million years old, which probably originated in the Oligocene epoch of the Tertiary period. Figure 2330 Rosids. Hibiscus. St. John's wort. Immature gourd fruit. Theobroma cacao, the source of chocolate. Flowers and fruits are borne on the trunk and large branches, not on twigs. The seeds are harvested, fermented, roasted, and ground into cocoa powder. Lupin. Mexican plum. Strawberry. Table 23-9 Representative families of the asterid clade. Figure 2331 In asterid flowers, such as Digitalis purpurea, all petals fuse to each other and form a tube. Only certain pollinators can fit into the tube far enough to obtain nectar, and inadvertently carry out pollination. Flowers of potato also have fused petals, but they are so open that we can see that stamens alternate with petals. Figure 2332 Ellagic acid is common in several eudicot clades but absent from one of the most derived, the asterids. Its presence deters many insects from eating the plants, but by approximately 50 million years ago, many types of insects had evolved a tolerance to it. Many asterids produce iridoid compounds such as this cornin. They are a relatively new class of chemical defense compounds, and thus, few insects tolerate them. Iridoid compounds may be partly responsible for the success of the asterids by keeping herbivory to a minimum. Figure 2333 One recently proposed cladogram of asterids. Figure 2334 Asterids. Potato. Milkweed. Goat's beard. The flowers of asters are really inflorescences, groups of many true, small flowers, called florets. Each outer petal is a ray floret, and each central flower is a disc floret. The dark cylinders are anthers that have fused around styles. The innermost disc florets have not yet opened. Flowers of Cavendishia show the derived feature of sympetally, petals fused together as one unit, except at their tips. In hydrangea, what appear to be large petals are really bracts located below the tiny flowers. Plants and people. Box 23-2 Vegetarianism, alternatives to eating meat. Our ancestors evolved to be omnivores. As a result, we modern humans can eat seeds, leaves, and fleshy roots because our flat molars allow us to grind and mash them before we swallow them, and our small intestine is so lengthy it ensures that plant matter remains inside us long enough to be digested. Similarly, we can eat meat because our incisor and canine teeth allow us to rip meat from bones, and our digestive system produces plenty of protein-digesting enzymes. However, just because we have the ability to eat meat does not mean we must eat it. A large number of people have adopted some form of vegetarianism to reduce the amount of meat in their diet or eliminate it altogether. There are several types of vegetarian diet. Ovo-lacto-vegetarians eat a diet based almost exclusively on plant products such as fruits, vegetables, greens, and seeds with some eggs and dairy products. The concept here is that animals do not have to be killed to obtain eggs and milk. For some vegetarians the only acceptable animal-based food is eggs, for others only milk-based foods, and for others even fish and seafood is acceptable. Vegans avoid all foods obtained from animals, including meat, eggs, and all dairy products. Strict vegans also do not wear clothing made from animals, such as leather belts, shoes, and coats or anything made with fur or turtle shell. A fruitarian diet consists of only fruits, nuts, and seeds, items that can be gathered without harming a plant. Fruitarians will not eat roots such as carrots, beets, and cassava because those can be obtained only by killing the plants. Flexitarians have a flexible approach to meat and other animal products in their diet. They eat mostly plant foods and merely reduce the amount of meat they eat. For example, flexitarians might set the goal of having meat-free lunches or designating several days of the week as being meat-free. The advantage of a flexitarian approach is that a person can succumb to the occasional hamburger or steak without feeling they have failed and might as well give up. There are several reasons for reducing or eliminating animal products in our diet and our daily lives. Many people do so for ethical reasons. They do not want to be responsible for animal suffering or death. There is a range of empathy here. Red meat is muscle tissue that can only be obtained by killing cattle. Cattle are mammals like we are, and they definitely feel pain and suffering. Obtaining milk from cows, goats, or other mammals does not involve killing them, so there is little or no suffering in many dairy operations. Gathering eggs from chickens or other birds also does not involve harming them, although some poultry farms keep the animals crowded together in poor conditions and clip their beaks so they won't peck each other. Eating fish and seafood involves killing the animals, but many people feel little emotional connection to fish, shrimp, and clams. Figure B23-2. This is an ovo-lacto-vegetarian meal because it contains eggs and cheese in addition to plant-based foods. A dessert of pie and ice cream fits into such a diet. When choosing your diet for ethical reasons, there are various issues to consider. Raising cattle in the United States and elsewhere is often cruel. Most cattle are raised on factory farms, crowded into large, dirty feedlots where the animals must stand in their own feces. They are given feed that includes antibiotics, growth supplements, and even the ground-up debris from cattle that were slaughtered earlier. When it is time to kill them, the cattle are poked with prods that give them an electrical shock, forcing them into a line into the bright, noisy slaughterhouse. Laws in many countries prohibit the slaughter of any animal that is not healthy enough to walk into the slaughterhouse, but that law is sometimes violated. PETA, People for the Ethical Treatment of Animals, has developed a set of guidelines for treating cattle more humanely, including giving them uncrowded pastures for grazing and safe food, and when they are to be killed the slaughterhouse should be quiet, dimly lighted, and with as few people as possible. The animals are still killed, of course, but at least their life and death were not miserable. For many people this humane treatment reduces their ethical objections and they will eat meat occasionally. To be certain the meat you eat has come from humanely treated animals, it is best to shop at reputable health food stores or organic food stores. Don't be afraid to ask about the treatment of the animals. Veal is especially objectionable because it is the flesh of calves that are killed while still so young they have never eaten anything other than their mother's milk. Often, the calves are kept in cages so small they cannot move because movement would cause their muscles to develop, making the meat less tender. The calves also may be kept in the dark to keep them quiet. It is difficult to justify treating animals like this just to have a particular kind of dinner. Eggs and milk do not require the slaughter of animals, and therefore many people do not believe it is unethical to consume these products. Here too it is best if the chickens and cattle are treated humanely and are allowed access to open pastures or pens with fresh air and water. People with the strictest ethical standards try to avoid all animal products, because they believe it is unfair for us to breed and maintain animals just for our own use. It is difficult, however, to eliminate the use of all animal products. Sugar is whitened by filtering it through the ash of animal bones, most cheese is made with rennet from calves' stomachs, clear juices and alcoholic beverages are clarified with gelatin from animal hides and bones, and so on. Other people avoid animal products for economic and ecological reasons. When we feed animals corn, soybeans, oats, and other feeds that we ourselves could eat, we are eating at a high trophic level. The plants are primary producers, the livestock animals are primary consumers, and we are secondary consumers. Typically, it takes 10 pounds of food at one trophic level to make 1 pound of food at the next level. Consequently, we need to feed cattle or chickens 10 pounds of oats just to get 1 pound of eggs or milk or meat. The other 9 pounds are lost as carbon dioxide and methane, urine, feces, and waste. If we ate the plants ourselves, we would need to farm only 1/10 as much land as we use now, producing much less sewage. It is estimated that as much as 70% of all the wheat, corn, and other grains we cultivate are used to feed livestock. Also, huge areas of tropical rainforest have been and are being cut down and converted into pasture for cattle just to produce hamburger. Reducing the amount of animal products we eat is an important aspect of a philosophy of living more simply, of trying to minimize the impact each of us has on our environment. An important consideration for anyone considering a vegetarian diet is whether it is healthful. The answer is easiest for a flexitarian diet. Virtually all studies show that diets low in meat and high in fruit and vegetables are much more healthful than ones with more meat. And it is now well established that even a strict vegan diet provides all the nutrients we need for a healthy life. We need to be especially careful to obtain adequate amounts of vitamin B12, which is not found in any plant but which can be obtained as a supplement or by consuming yeast products. Many people believe that athletes and bodybuilders need a high protein intake and must have meat, but that has been shown repeatedly to not be true. Most of us Americans consume much more protein than we need, and the plants of a vegetarian or vegan diet provide adequate amounts for anyone. We must be careful to obtain adequate amounts of all 8 essential amino acids. All plants, animals, fungi, and other organisms construct their proteins from the same 20 amino acids, and we humans can synthesize 12 of them within our own bodies, but our metabolism cannot make 8 of them. Dairy and egg food supply adequate amounts, and so do the proteins of soy, amaranth, quinoa, and several other seed plants. The proteins of many food plants such as wheat, rice, beans, chickpeas, and others have adequate amounts of 18 or 19 amino acids but only sparse amounts of 1 or 2 others. We can avoid having a deficiency by eating what are known as complementary foods, foods in which one member has plenty of an amino acid that the other member lacks, and vice versa. For example, a combination of brown rice and beans, beans and corn, tofu and rice, or one of hummus and wheat pita bread provides adequate amounts of all 20 amino acids. Think about a typical Mexican meal that includes beans and corn tortillas. These two plant foods together provide all essential amino acids. The same is true of a typical American lunch. A peanut butter sandwich provides complementary proteins. The take-home message here is that we should eat less meat and animal products. Both our bodies and our environment will be healthier. Figure 2335 Hymenoxin is a sesquiterpene lactone, a member of the chemical arsenal of asterids. This particular compound occurs in bitterweed of the southwestern United States. The plant is eaten by sheep only during droughts when no other plants are available. Sesquiterpene lactone causes hemorrhaging of all internal organs of the sheep. Many members of Asteraceae contain secretory canals lined with cells that produce a variety of toxic compounds. This canal is in the style of sunflower. At the next level. 1. Focus on a monocot group. The angiosperms are such a large, diverse group that it is difficult to become familiar with all of them unless you spend your entire life studying them. Fortunately, you do not have to know them all. But I do recommend selecting a particular group and studying it at the next level. I recommend the elismatales because these are unusual. Most have become adapted to living in water and many could be mistaken for mosses or algae until you look closely. You already know a great deal about typical angiosperms. The elismatales will surprise you at how extreme angiosperm modifications can be yet still be understandable. 2. Focus on a eudicot group. For this I recommend the rose family, Rosaceae. You already know many of its species, so read even more about these. You know apple fruits, but are you familiar with the tree? You know strawberry fruits, but what about the plant? Would you ever guess that an herbaceous strawberry plant is related to a woody apple tree? This family has evolved by natural selection to have many growth forms, and we have applied artificial selection to alter the flowers, fruits, and vegetative bodies even further. 3. Wheat, rice, corn, grasses keep us alive. The grass family Poaceae has hundreds of species, and many have been domesticated as foods. Wheat, rice, and corn are familiar examples of plants we rely on. Almost every person on Earth eats one of these grasses every day. There are many other grasses that have been domesticated as well. Research the species we cultivate and learn about the benefits and problems of each. Why do we cultivate more wheat than rye? We probably drink more barley than wheat. Why? Summary. 1. All flowering plants, members of division Magnoliophyta, have closed carpels, long pollen tubes, double fertilization, and, with very few exceptions, sieve tubes and vessels. 2. Division Magnoliophyta is almost universally believed to be monophyletic. All of its evolutionary lines can be traced back to a single common ancestor. The earliest fossils of leaves and wood date to approximately 125 to 130 million years ago, and fossil pollen from earlier periods has been found. 3. The earliest angiosperms may have been weedy shrubs that grew in areas not conducive to fossilization. 4. Early characteristics of angiosperms are believed to be flowers with many sepals, petals, stamens, and carpels arranged in spirals, uniaperturate pollen, radially symmetrical flowers, insect pollination, and wood without vessels. Later, tracheid-like vessels with scalariform perforation plates arose. Early angiosperms are believed to have been perennial, none was annual or herbaceous. 5. Some major evolutionary modifications that have occurred in many groups are reduction in the number of each flower organ, imperfect flowers, bilateral symmetry of flowers, nectaries and fragrances that attract only specific pollinators, wind pollination, various seed or fruit distribution mechanisms, biennial or annual herbaceous habit. Wood with shorter, wider vessel members, larger amounts of wood parenchyma, and numerous types of anti-predator compounds, perhaps 2 or 3 generations of these. 6. division Magnoliophyta contains basal angiosperms, monocots, and eudicots. 7. Monocots can generally be recognized by their long, strap-shaped leaves, flower parts in threes, parallel leaf venation, numerous vascular bundles in the stem, not arranged in a ring, and no ordinary secondary growth. 8. Eudicots can generally be recognized by broad, not strap-shaped leaves, reticulate leaf venation, flower parts in fives or fours, but not threes, pollen triculpate or triculpate-derived, vascular bundles in the stem arranged in one ring, and woody growth or an annual or biennial herbaceous body. 9. In the classification presented here, monocots contain several orders in the commelinoid clade, and eudicots contain basal eudicots, rosids, and asterids. Important terms. Angiosperm carpels anthocyanin pigments asterid clade basal angiosperms. Betylene pigments campanulid order closed carpel commelinoid monocots division Magnoliophyta double fertilization eudicots fabids. Generalized flower iridoid compounds Lamiid order Malvids monocots perisperm pistil. Primitively veselless Ronnaline flower rosid clade secondarily veselless septal nectary sympatally triculpate uniaperturate zygomorphy. Review questions. 1. The earliest fossils clearly recognizable as parts of flowering plants are in rocks more than million years old. 2. What transformations must have occurred if gymnosperms really were the ancestors of angiosperms? Which might have occurred earlier and which later? 3. If someone were to examine the stamens of most flowering plants, they would never automatically think of them as sporophylls because they have very little leaf-like structure, but look at the 4 stamens in figure 23:1. These are from flowering plants. What leaf-like characters do they have? Are they flat and at least somewhat resemble the blade of a leaf? Do they have vascular bundles that branch, at least somewhat resembling the reticulate venation of a dicot leaf? Most dicot leaves have 3 vascular bundles entering their petioles. Which 2 of these stamens also have this leaf-like character? Which 2 have only 1 vascular bundle? 4. During fertilization in flowering plants, one sperm cell nucleus fuses with the egg nucleus, producing the diploid zygote. The other sperm nucleus fuses with producing the triploid, which develops as temporary tissue that nourishes the embryo. This process is called fertilization. What tissue nourishes the embryo in gymnosperm seeds? 5. Why were many modern genera of dicots initially thought to have come suddenly into existence about 130 million years ago? Why do we now think that those leaf fossils do not really belong to the modern genera they were named for? 6. What is meant by a closed carpel as opposed to an open cone scale? 7. Describe what the early angiosperms may have been like with regard to flowers, body, ecology, wood, and pollen. For each of the characters, what living angiosperms still have those characters? In what ways have those characters changed in certain lines of evolution? 8. Concepts about the nature of the early angiosperms have changed. In the last century, members of the subclass Hamamelidae were considered the most relictual of the living flowering plants. What kind of pollination do these plants have? Insect pollination, wind pollination, or bird pollination? Give the common names of 4 members of this group. Although these plants are woody trees like many conifers, their wood has 3 characters that wood of conifers does not have, so we no longer think that hamamelids are the most relictual flowering plants. What are the 3 characters? 9. About 100 years ago, C.E. Bessey developed the hypothesis of the Ronnaline flower. What are some of the characters of this type of flower? Is the Ronnaline flower wind-pollinated or insect-pollinated? 10. Do most botanists believe that the flowering plants are monophyletic, or do they believe that they are actually an unnatural group consisting of several lines of evolution that do not share a common ancestor? 11. Describe ambarella and water lilies. On the basis of morphology, would you conclude that they are closely related? What evidence is used to support the hypothesis that they are ancient lineages from basal angiosperms? 12. All plant families have a name that ends in ace, and some families also have old names, still commonly used, without this ending. The palms are a family with two names. What is the new name that ends in ace, and what is the older name? What kind of a compound leaf do the feather palms have? The fan palms? Why are palm flowers so seldom seen by most people? 13. Philodendron is a plant you may know. They and their close relatives are characterized by a distinctive inflorescence. It has tiny flowers embedded in a thick stalk called a, and it is surrounded by a large bract called a. 14. What is the name of the grass family? About how many species does it contain? Name 7 genera of grasses that are used for food. What kind of pollination do grasses have, animal or wind? 15. Zingiberals contain some of the most familiar of all houseplants. Name several of these. 16. Liliaceae contains many familiar plants. Name 6. Which closely related family contains the irises? Which contains yams? 17. The Orchidaceae is the largest and most diverse family of monocots. What is the most common orchid habit, being an epiphyte or being terrestrial? Are orchid flowers bilaterally symmetrical or radially symmetrical? Describe orchid seeds. 18. Certain eudicot clades contain plants with many relictual features. Name and describe several. 19. Cor Caryophyllales contains many familiar plants. Can you list 4 members of this group? The group is unusual in having pigments rather than anthocyanins in their flowers. Another feature of this group is that endosperm develops little or not at all, and instead, the seeds have. Which develops through proliferation of cells of the. 20. Give the name of the families of each of the following: A. Camellias, B. Chocolate, C. Cotton, D. Violets. E. Pumpkins and watermelons, F. Cranberries and blueberries. 21. One important character in rosids is the presence of leaves. The subclass is named for the rose family, Rosaceae, and this one family has many genera that produce fruits that we like to eat. Name the genera of the following fruits, all in the rose family: A. Apples, B. Almonds, apricots, cherries, peaches, and plums, C. Strawberries, D. Blackberries and raspberries. 22. Textbooks often use cacti and euphorbias to illustrate convergent evolution because many euphorbias have globular, succulent bodies that strongly resemble those of cacti, yet the two families are not closely related. What are the names of the euphorbia family and of the cactus family? Which major clade is each in? 23. Name the families that contain the following species: A. Sunflowers, dandelions, and daisies, B. Milkweeds, C. Potato, tomato, and tobacco, D. Mints and lavender, E. Coffea. 24. Think about the Coniferophyta and the Magnoliophyta of this chapter. Which has more families, genera, and species? Which has more diversity of body types? Which has species in the greatest number of habitats? Which group originated earlier in evolution, and which originated later? The older group has had more time to diversify. Is it the more diverse of the two? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Chapter 24. Ethnobotany, Plants and People. Chapter opener image: People depend on plants for food, clothing, lumber, and many other things. But we also rely on plants to help us emotionally. Here they are providing a refreshing, calm location in which students relax between classes. Plants are often used as symbols for strong emotions we have difficulty expressing. They speak for us at funerals, weddings, when friends are ill, and also just as a sign of friendship. Outline. Concepts. Food plants. Plants that supply carbohydrates. Plants that supply proteins, oils, and vitamins. Plants that provide flavor, herbs and spices. Plants that provide drugs. Plants that provide fibers, wood, and chemicals. Fibers. Chemicals. Wood. Box 24-1 Plants and People, the Domestication of Plants. Box 24-2 Botany and Beyond, Lipids, Oils, Fats, Trans Fats, and Human Health. Box 24-3 Plants and People, Natural Drugs, Endangered Species, and Women's Rights. Learning Objectives. After reading this chapter, students will be able to discuss the times and places in which early humans began to domesticate crops. List 6 plants that provide carbohydrates. Describe several human uses of plants that produce proteins, oils, or vitamins, and know the difference between complete and incomplete dietary proteins. Describe the plants that supply us with chocolate, vanilla, coffee, tea, pepper, and spices. Identify plants that produce either harmless or harmful drugs, including tobacco and marijuana. Explain how we use plant fibers today and describe new alternative plants that may provide fibers while causing less harm to the environment. Did you know? People changed from being hunter-gatherers to farming city dwellers only after the end of the Pleistocene glaciation 12,000 years ago. Aztec chocolate was savory and bitter. Sweet milk chocolate was invented in Europe. The quest for spice plants such as cinnamon, cloves, and black pepper was the motivating force for the discovery of the Americas, the circumnavigation of the Earth, and virtually all other early worldwide explorations. Evidence continues to accumulate that marijuana has many beneficial effects and virtually no harmful ones. Concepts. Ethnobotany is the scientific study of relationships between plants and people. The science is new, the relationships are ancient. We humans have relied on plants during our entire existence. Some of our teeth, the incisors and molars, are adapted for biting off and grinding plant parts such as leaves, fruits, seeds, and stems, whereas our canines are adapted for tearing animal flesh. We are omnivores. Food plants have been and still are crucial to our survival. Our ancestors also relied on plants for shelter and protection, especially forest trees, at least as far back as 125,000 years ago, perhaps even much farther. Our ancestors learned to use fire, and then to control it, how to burn wood to keep themselves warm and scare away predators. They learned to roast meat and much later, after pottery was invented, learned to boil food. Even today, almost half the world's population still relies on wood fires for warmth and cooking every day. Throughout our entire time as a species, Homo sapiens in the family Hominidae, Earth was in the Pleistocene Ice Age, also called the Pleistocene Epoch. Snow and ice covered much of the northern hemisphere and all mountainous regions everywhere. The lowlands of Africa were mostly free of ice and snow. It is there that we humans diverged evolutionarily from our close relatives, and where we hunted animals, gathered berries, seeds, and firewood. About 12,000 years ago, a momentous event occurred. Earth's climate became warmer, glaciers retreated, and plants and animals spread into vast regions previously covered by ice sheets. The Holocene epoch had begun. It is the warm period in which we are living now. The Holocene is probably an interglacial period, the Ice Age. Probably has not ended, we are merely experiencing one of its temporary, cyclical warm periods. The dramatic climatic warming that occurred in the last 12,000 years could have been disastrous for us, but instead we adapted in a remarkable way. We began to cultivate some of our food rather than merely searching for it. No other animal does this. Our ancestors saved part of their harvest and planted it the following spring. They protected their fields, and they chose to cultivate plants with desirable characters such as larger fruits and seeds while weeding out those with less desirable traits. Thousands of years before Charles Darwin and Alfred Russel Wallace discovered natural selection, our ancestors were practicing artificial selection and the domestication of plants and animals. As far back as 11,000 years ago, some early people in South America started cultivating potatoes. Perhaps the very first crop plant, wheat and lentils, were domesticated 9,000 years ago in the region that stretches from southeastern Turkey to western Afghanistan, an area now known as the Fertile Crescent. Not long afterward, rice farming began in Southeast Asia, and later pearl millet was domesticated in Africa. Wherever people were in the world, they began cultivating plants shortly after global temperatures began to rise, and agriculture was invented at least 4 separate times on different continents. Figure 24:1 For 50 million years, lasting until just 12,000 years ago, much of the Earth looked like these mountains near Anchorage, Alaska. At that time, the snow and ice would have been thicker everywhere, and most lakes and even major portions of the oceans would have been covered with a sheet of ice. The close relationship that developed between people and their crops had profound consequences for us. Archaeological studies of the Fertile Crescent region reveal some of those consequences. Crops have to be defended, so people settled in fertile areas and stopped migrating. They built villages, and then very soon the first cities came into existence: Ur, Sumer, and Babylon. Erbil, in modern Iraq, was already a city 8,000 years ago. People learned to live together and govern themselves in groups larger than just family units or small tribes. Writing was invented to keep track of the contents of sealed containers of food, wine, and beer. Mathematics was developed, especially the geometry needed to survey cropland and build canals and irrigation projects. The Holocene warming and the development of civilization changed our relationship with many plants other than those that provide food. Cotton, flax, and hemp were grown for their fibers. Medicinal plants were tended in drug plant gardens, and with the discovery of copper and then bronze, sharp, durable axes and saws could fell trees for the wood needed to build temples, palaces, and ships. Of special significance, one of the first things early civilized people did was to cultivate plants for their beauty. The Hanging Gardens of Babylon are perhaps the most famous but were not the first. Much early Egyptian art depicted ornamental plants. In the thousands of years since people began to cultivate plants, agriculture has changed greatly, but our reliance on plants has not. This chapter describes only a few of the many plants that are essential to us. You will undoubtedly notice others as you go about your life from day to day. Food plants. Plants that supply carbohydrates. Carbohydrates are crucial to our lives. Our bodies use sugars and starches in many ways. Some are respired and produce adenosine triphosphate, others are building blocks of our own carbohydrates, and many are chemically modified and used to construct important molecules such as amino acids, lipids, nucleic acids, and many others. We cannot digest carbohydrates such as cellulose, hemicelluloses, and lignin, but these provide bulk and keep our digestive systems moving material along regularly. The greatest part of our diet, in terms of weight, volume, and calories, is usually carbohydrates. We eat bread, pasta, pastries, potatoes, and rice, and all consist mostly of carbohydrates. Figure 24:2 Several thousand years ago, the region of the Fertile Crescent received more rain than it does now, and large areas could be cultivated. Foods rich in carbohydrates come from several plant families, but the grass family Poaceae is especially important. All our cereal grains are the small fruits of grasses, each with one seed: wheat, rice, corn, oats, rye, barley, and more. If wheat and rice were to suddenly disappear, almost the entire human population would starve. The fruits of grasses are technically known as caryopses, but we call them grains, kernels, or seeds. Each has a tiny embryo and a large amount of endosperm, which is almost entirely starch, which is easy to digest into its monomers, glucose. When dry at harvest, cereal grains are stable and can be stored for years if kept dry and free of mice and similar animals. Cereal grains are easily prepared as food simply by boiling, or by grinding them into flour that can be made into bread or pasta. All can also be fermented to beer, sake, or whiskey. The seed coat and fruit wall together of each caryopsis are called the bran. If the entire kernel is ground to flour, we obtain whole wheat flour. For white flour or all-purpose flour, kernels are shaken to knock off the bran, and grinding produces a finer, smoother, more uniform white flour that is preferred for cakes and more elegant dishes. But removing bran also inadvertently removes the embryo as well. The embryo contains enzymes that digest the endosperm, so grinding the whole grain with the embryo disperses digestive enzymes throughout the flour. Unless it is kept perfectly dry, this whole wheat flour undergoes self-digestion and becomes rancid. Removing the embryo before grinding produces a flour that can be stored for years with little change in quality, but which is lower in proteins, vitamins, and minerals. Recently, more appreciation is being given to more natural foods, foods with fiber, and foods that have been processed less. It is becoming more common to see whole wheat bread and pasta. Figure 24:3 wheat fields can be large or small, irrigated or dryland. Wheat thrives in temperate climates with moderate temperatures and with moderate amounts of rain. It does not grow well if summers are cool and damp, or in tropical areas that are too rainy. Figure 24:4 Each stalk of modern wheat produces several rows of large grains. These shown here are immature. They still have the stamens attached, although by this time the carpels have been fertilized and the seeds are developing. Each grain is an individual fruit, each containing one seed. When the wheat is mature, it will dry out and turn tawny brown. Figure 24:5 The fruit of any cereal grain will contain a small embryo and a large amount of endosperm, both surrounded by the dry fruit. Wheat, which now exists as several species, is an ancient crop, having been cultivated at least 9,000 years ago in the Fertile Crescent. Einkorn wheat probably resembles the first domesticated wheat. It is a small plant that grows even on stony ground, has only a few grains per plant, and is diploid. It still grows in Turkey in marginal areas not used for cultivation. Einkorn wheat now is cultivated mostly for its historical value and to maintain genetic diversity. Emmer wheat is a later wheat, and it too is a small plant with few kernels per plant, but it is tetraploid. Emmer wheat was cultivated by ancient Egyptians and supported not only their empire but also that of the Romans. We are not exactly certain when and how our modern wheat, Triticum aestivum, originated. It is hexaploid, with 6 full sets of chromosomes in each nucleus. Unlike the other two, T. aestivum is a large plant with many kernels per stalk. Figure 24:6 Until recently, one of the problems with wheat was that its shoots grew to be too long and weak, and would often fall over due to wind or strong rain. This made it difficult to harvest, and the lodged wheat might rot if it lay on wet soil. Figure 24:7 A new genetic modification of wheat was to alter it such that its stems are shorter and stronger, and thus they almost never lodge. This allows all the wheat plants to stand upright until the fruits are mature, then when they dry, they can be harvested easily. Wheat differs from other cereal grains, and from all other starchy foods, in that it contains significant amounts of the elastic protein gluten. As wheat dough is kneaded, gluten molecules are stretched into springy strands within the dough, which trap carbon dioxide bubbles produced as yeast respires, which causes the dough to rise. By kneading wheat flour to develop gluten strands and then allowing the dough to rise before baking, we get a bead that is soft, easy to chew, and more readily digestible. If flours of rice or corn, which have no gluten, were kneaded with yeast the same way, nothing would happen. If allowed to set, the dough would not rise, the carbon dioxide would just escape. Rye bread, corn bread, and others have significant amounts of wheat flour or gluten mixed into the dough. When making cookies, cakes, and biscuits with wheat flour, the dough must not be kneaded if elasticity and stringiness are to be avoided. Rice was domesticated in East Asia about 7,000 years ago. It is now the main source of carbohydrates for more than half the world's population. The two most common varieties of rice are Oryza sativa subspecies sativa, which is moist and sticky when cooked, making it easy to eat with chopsticks and to mold for sushi. Oryza sativa subspecies indica is fluffy and drier when cooked, and is the main rice used in the West. To produce rice that is white when cooked, it is polished to remove its bran, and vitamins are lost in the process. People who rely on rice as their main food with sparse vegetables and little meat may have a lack of vitamin A, which results in vitamin A deficiency disorders, including impaired vision, compromised immune systems, and many other symptoms. Genetic engineering has produced golden rice, which is rich in beta-carotene, the precursor of vitamin A. Golden rice could greatly improve the diet in many parts of Southeast Asia, but concern over its being a genetically modified organism. Food has limited its acceptance. Natural rice has genes to produce beta-carotene, but they are expressed in the leaves, not in the seeds. Thousands of varieties have been screened, and none has the precursor to vitamin A in the seeds. It seems that golden rice, a GMO, is the only way to make rice more nutritious. Other cereal grains such as oats, barley, rye, and corn are consumed in much smaller quantities than wheat or rice. We eat them as oatmeal, rye bread, and sweet corn. Much of barley is brewed into beer. But the greatest amount of these is used as feed for livestock such as cattle, pigs, poultry, and sheep. These animals are raised to supply us with meat, milk, or eggs. If we were to eat the grains directly, we would be primary consumers, but because we consume the animals instead, we are secondary consumers. We are eating higher on the trophic pyramid. Generally, this means it is necessary to use 10 kilograms of cereal grains to obtain 1 kilogram of animal protein. If we would consume less meat, we would not need to convert so much natural land to farms and ranches. Oats are high in protein and beneficial lipids, and they help keep cholesterol at safe levels. Oats were domesticated as recently as 3,000 years ago, and at present they are becoming more popular in our diets because of their healthful effects. Barley is an ancient crop, having been domesticated about 9,000 years ago. The ancient Sumerians, the people who built the first cities and irrigation projects in the world, discovered how to brew barley into beer. This was a momentous discovery because fermentation produces more vitamins, making beer more nutritious than raw barley, and it sterilizes the beer by killing microbes that grow on the grain and in the water. The yeasts that ferment beer use only glucose, not starch, so before brewing begins, the barley is lightly roasted to convert starch to glucose. This toasted barley is called malt, and it is not only used for brewing, but also in malted milkshakes. Figure 24-8 rice is a cereal grain that greatly resembles wheat in size, shape, and habit. This is grown in Louisiana, East Texas, and California where land is extremely flat and large areas can be flooded for irrigation. Figure 24-9 oats that are ready to be harvested. Each stem produces many fewer grains than does wheat, and oats are not as rich in protein. The stems that remain after the grains are harvested are referred to as straw, and are used as bedding for livestock but have virtually no nutritional value. Figure 24-10 beer is brewed in large vats that allow sugars in toasted barley to ferment, which converts them to ethyl alcohol. The vats are open, allowing carbon dioxide to escape. The bubbles that you see when you pour a beer are carbon dioxide that has been added artificially just before the beer was shipped to market. Corn was domesticated in Mexico as long ago as 9,000 years before present. Corn caryopses are important in the cuisine of Latin America and the United States, being ground into gluten-free flour and eaten as corn tortillas. cornbread, whole kernel corn, and corn on the cob. Today, much of the crop of corn kernels is processed into corn syrup and used as a sweetener, as animal feed, or converted to ethanol. The entire corn plant can also be chopped into pieces, piled into large mounds and allowed to partially ferment. This produces a slightly alcoholic animal feed called ensilage or just silage. Cattle love it. Other plants besides cereal grains also provide significant amounts of carbohydrates. Potato tubers grow underground at the base of herbaceous annual plants. Each plant produces many tubers. The above-ground portions of the plant's